Hi everyone. Let's discuss today briefly on membrane transport. What is membrane transport and why is it important? Membrane transport is nothing but the movement of molecules across the cell membrane. Why it's important? So because the peculiar feature of cell membrane, that is semi-permeable cell membrane. And what are the things has to be transported? We know that we usually will take food and it has to be absorbed, and the absorbed product, it has to be transported to the various site of action. Also, if there is a signal, we have to release the neurotransmitters and it has to be transported to the site of action, or the stimulants for the synthesis or release of hormones. All this has to be transported from one place to another. So, what are the main things in this membrane transport and why is it important? Let's come in detail. We all know that cell membrane is made of lipid bilayer and it has proteins and lipids in that, and mainly lipids are glycerophospholipids, sphingolipids and steroids, and proteins what are seen as integral protein, that means the protein seen through the membrane are passed through the membrane, then peripheral protein that are attached to the surface, and amphiprotic membrane which are present both in cytosol as well as in the membrane. And main important thing, cell membrane is semi-permeable. And what all things has to be transported across the biological membrane? The exchange of molecules and ions in and out of cell wall, and also the components has to be transported from nucleus, endoplasmic reticulum and mitochondria. Then example for transported things include glucose, sodium, potassium, calcium, ATP, mRNA, etc. The cell membrane is permeable to water molecule and small uncharged molecules like oxygen, carbon dioxide, but impermeable to large and polar molecules because of the particular feature of cell membrane. Here the transport of such impermeable compounds occur through carrier proteins, channels and pumps within the membrane. Transport may be active by utilizing carrier proteins, utilizing the energy source, or it may be passive by simple diffusion or through facilitated channels. We can see the permeability of molecules across membrane. Small uncharged molecules like ethanol, they are permeable to the membrane. Then gases like carbon dioxide, oxygen, nitrogen, they are permeable to membrane. But polar molecules like urea, water, they are slightly permeable, and the large uncharged molecules like glucose, fructose, they are impermeable. Then ions, positively and negatively charged, they are impermeable. Then polar charged molecules are also impermeable to the cell membrane. So these slightly permeable and impermeable molecules, it has to be transported from one side of the membrane to other side to perform various physiological functions. And that is the importance of membrane transport. And main force that involves the transport of molecules is diffusion. That is the net movement of molecules from one side from higher concentration to lower concentration. And the cellular transport includes passive, that is purely diffusion driven and also follows the concentration gradient. Then facilitated, that is aided by means of special protein and also follows the concentration gradient. Active transport, it is protein guided but against the concentration gradient, so it requires energy for the transport. And there are two classes of transport, that is transport by channels usually seen in diffusion, and by means of carriers or transport molecules that are also seen in facilitated diffusion and active transport. This is a diagrammatic representation for the passive transport. Passive transport includes simple diffusion, that is movement of molecule from higher concentration to the lower concentration through the semi-permeable membrane, or it requires a transport molecule for for the transport of, and here also it's from higher concentration to the lower concentration. The movement occurs through pores which are created by ion channels or porins, and they open and close in response to a particular stimuli. And passive transport usually occurs within different systems. Example are movement of oxygen through membrane, movement of glucose into the cells, movement of oxygen in and out of, water in and out of the cells, formation of kidney filtrate. They are all example of passive transport. And the energy here, that is by means of kinetic energy. Facilitated diffusion is a type of passive transport. Here energy is not required, but it is held by means of integral membrane proteins. That is a spontaneous passage of molecules across the membrane with a specific transmembrane protein. An example is glucose transporter of GLUT family, then amino acid transporters, urea transporters, etc. Let's discuss in detail on GLUT transporter. Among the glucose transporter, there are several types of GLUT transporter and also active transporters. The GLUT1 transporter, that is a unipolar protein and helps in the transport of glucose molecules across the plasma membrane of mammalian cells. This unipolar protein means this type of transporter, it allows the transport of a single type of molecule. It is usually seen in the red blood cells and is responsible for glucose uptake required for the respiration in all cells. We know that blood cells are rich in glucose content, or blood plasma has higher concentration of glucose. So since it is having higher concentration of glucose, here for the transport of these glucose molecule, it doesn't require an energy. So facilitated diffusion is more suitable, and it can be done by means of a gated pour mechanism. It is simply like opening a gate and closing a gate and releasing the contents. Here we can see this is the outside cell and this is the inside cell. So when glucose molecule comes in contact to the glucose transporter, GLUT1 transporter seen in the blood cells, erythrocytes, here there is a conformation change so that the transporter is open so that glucose can enter inside. After the transporter entering inside, there a conformation change so that the entry is closed, and now the conformation change occurs such that the opening is towards the other side of the cell membrane. So glucose, whatever it is seen in the outside cell, that is being passed to the inside of the cell by means of this gate mechanism. So let's discuss on active transport. There are numerous situations in the living cells where the molecule pass across the cell membrane from an area of lower concentration to an area of higher concentration. Please note the lower concentration to higher concentration. Usually this doesn't occur by means of diffusion. So there occurs there it needs energy source for this type of transport, and it is called an active transport. Here work must be done to overcome the influence of osmosis and diffusion. An example of active transport is the maintaining the levels of sodium, potassium and calcium in our body cell. Here we can see the outside cell that is having lesser amount of potassium, but inside cell that is having higher amount of potassium, but still it requires the transport of potassium from outside to inside, and that is against concentration gradient, so it is an active transport. Here also same situation, from inside to outside, the transport of sodium. Here that is higher concentration, that is against concentration gradient, again an active transport. The calcium also from higher to lower concentration. So against from lower concentration to high concentration. So still again against concentration gradient. Active transport may draw energy from the hydrolysis of ATP. Usually that is seen in many of the transport mechanism. A very good example is the sodium dependent transporters by the absorbance of light, that is usually seen in photosynthesis. by the transport of electrons usually seen in the electron transport chain, that is the proton motive force created is responsible for the synthesis of ATP, or coupling with other processes that are moving particles down their concentration gradient. Example for active transport is movement of ions across membrane, neurotransmitter secretion, white blood cell phagocytosis, absorption by intestinal cells, hormone and cholesterol uptake, hormone regulation, intracellular trafficking of molecules. Here, all cases it requires the energy source ATP. This is the model for the active transport. Here we can see the molecules. This is the amino acid, and amino acid is transported along with the sodium. So, if the amino acid is transported, this is the amino acid. Amino acid is transported from inside cell, that is having low concentration, to the higher concentration, and the sodium is transported from inside from extracellular fluid to the inside of the cell. So these two movement of two molecules are opposite to each other, and the movement of these two molecules are opposite. They are called antipode system. Here this movement requires the energizing power ATP, and in this case amino acid and the sodium both are moving towards the inside of the cell, and both are in same direction, and that is called the sympod mechanism. So in active transport, there may be antipode mechanism where the movement of molecules in opposite directions, or sympod mechanism, movement of molecule in same direction, or unipode mechanism that requires movement of only one molecule. Primary active transport uses ATP. That is, an example for this is sodium potassium pump. Here this pump operates by the energy expenditure of ATP. Secondary active transport is also there that requires the electrochemical gradient for the transport of molecule. It is also called a coupled transport or co-transport, and the example is ATP synthase. An example for active transport is uptake of glucose in the intestine. Earlier we had discussed on the regarding the transport of glucose in the blood cells. Now we here that is the uptake of glucose in the intestine. We know that the intestine is having lower concentration of glucose compared to the blood cells. So from the intestine it has to be transported towards the blood cells. This we had discussed based on the co-transporter. There are uniporter, sympod and antipode mechanism. And in humans, sodium is usually co-transported across plasma membrane whose electrochemical gradient is used to power the active transport of a second ion or molecule against its concentration gradient. This is a glucose transport mechanism in the intestine. This intestinal villi, it will absorb the digested material, and from there it has to be transported to the blood capillaries. So it requires it is against a concentration gradient. That means here it is having lower concentration, but the blood cells are having higher concentration. So glucose from here it is transported by means of this transporter. This is called the SGLT. That means sodium dependent glucose transporter. So transported along with the sodium, and that transport works by means of this sodium potassium ATP. So this energy, ATP hydrolysis energy, drives this transport from the intestine to the capillaries. We can see it here. These all are regarding the membrane transport. That is, transport is of mainly two types: passive transport and active transport. In the passive itself, there is simple diffusion and then facilitated diffusion, that is aided by means of membrane proteins. Then active transport means that also aided by membrane protein, but it requires energy since it is against the concentration gradient. So let us discuss on Donan membrane equilibrium. That is a special effect attributed in the membrane transport or membrane exchange of molecules within the membrane due to the presence of an impermeable large molecule. When two different concentrations of electrolyte solutions are separated from each other by a semi-permeable membrane, and if a non-diffusible ion is present, non-diffusible ion, one example is protein, in one side of the membrane, then there is an unequal distribution of diffusible ions between the compartments. This unequal distribution of ions is to maintain the electrical neutrality, that is, charges on both sides of the membrane has to be made equal. And this behavior of charged particle is called Gibbs-Donan equilibrium. We can see what is, I will describe here. Here this is cell membrane and this is the extracellular space and this is the cytoplasm. We can see here the chloride content in the extracellular fluid, that is more than in the cytoplasm. So usually we will expect that this chloride, it can move to the cytoplasm by means of simple diffusion. But there occurs a problem that there is a negatively charged protein and chloride is also negatively charged. Due to this negatively charged protein, it will inhibit the free movement of this chloride towards the cytoplasm. And here what is of main consideration is electrical neutrality. That means how much our protein is having the charge, only that much balancing amount of chloride can be transported from extracellular to the intracellular space. Main thing, we have to maintain the electrical neutrality here. Not simply movement of chloride based on the equilibrium, but the movement of molecule based on the electrical neutrality. Due to Donan effect, the negative charge of non-diffusible anion, that is protein we had seen before, hinders the diffusion of cation, that is the diffusion of cation from A side and favors the distribution of anion from the B side. Hence, chloride diffuses from B side to A side down its concentration gradient along with some potassium due to its opposite charge. At equilibrium, more osmotically active particles are present on A side than on the B side. Therefore, osmotic pressure on A side is greater than B side because of the osmotic pressure osmotic pressure depends on the number of ions. So, the Donan equilibrium has mainly different effects on the human body, that is proteins present inside the cell due to Donan effect. More osmotically active particles are present in the cells than in the interstitial fluid. Sodium-potassium pump counterbalance the effect. Due to the difference in the concentration of diffusible ions, an electrical difference is formed across the membrane, which is balanced by the opposite charges on the membrane. There are more proteins in the blood, due to which ionic movements occurs across the capillary walls. This Donan equilibrium is having many applications. Reverse chloride shift, then chloride shift are also application side of this Donan membrane equilibrium. Only one thing to note down, in Donan membrane equilibrium, the movement of molecule is based on electrical neutrality, not based on simple colloidal, colloidal property. Based on not a colloidal property, it is due to the colloidal nature of the protein, but it is simply maintaining the electrical neutrality among both sides of the membrane. Hope you all followed this session. Thank you.